I got you. I got to do a little picture here before I forget.
And I gotta add the PowerPoint before I go to call up to do other stuff.
Chaser, you were perfect. A little nappy poo, that's pretty good. Yeah,
Oh my god.
She did perfect. Okay.
Alright, like a
Okay. Madison, tell us a little bit about Taser.
She looks like a German Shepherd, but
Yes.
she's like 100% personality husky, as you probably just heard. I adopted her over 2 years ago. She's 3. Before the rescue I adopted her at, she was actually only given like 72 hours to live before she was going to be euthanized.
Oh, I see. She was on
Yeah.
the list of euthanasia
Yeah.
to be put down. Yeah.
But no, it's got it. It's named after Jonathan Taze. That is.
Named after who?
Jonathan Taze. He's the Black Hawks captain.
Oh, okay. Okay. Excellent.
That's about it. She's a big
3 years
girl.
old, and
Yeah.
any health problems?
She has like really bad seasonal allergies. That's about the only thing.
Okay.
Itchy skin.
She looks like she's pretty good. How long has she been here? I mean, she looked like she was very comfortable. From the
We
get-go?
got here at like 30.
Okay, so that's not that long ago.
Yeah, she makes herself very comfortable.
That's great, yeah. Because, we always, if I'm running late, the dog comes in, and I should tell the TAs too, if you feel uncomfortable with the dog, we would always say, ask the owner to maybe leave, you know what I mean? Because for the safety of everybody and for the safety of the dog, but, so always use your best judgment. And, one thing I'm trying to teach you how to interact with the dog. And here's one thing I haven't said, and some people do this, when they meet a strange dog, especially a big one, they want to put their face right down by the dog. I think you've seen that, where people will put their face down there. And that's, that's the wrong thing to do, because in the big dog world, if you stare somebody in the face, if you're a 2-dog, remember I was a dog in a former life, so I know this. If you stare a dog in the face, that's a challenge. That's a challenge to me, okay? So you never stare a dog in the face, and it's always good, big or small, let the dog come up to you. If it doesn't want to interact with you, then, there's all kinds of other dogs, and it's just sometimes they don't like you or a man or a woman or a certain color of hair or whatever hat. You just got to live with it. It's hard for me to say that because I want to go and hug every dog, but I especially the ones that are like the seeing eye dogs and stuff, I have to like resist because they're working. You're not supposed to interact with them at all, and that's really hard for me. So yeah, I've caught myself many a time. Okay, well, any questions? Any burning questions on endocrinology before I get into my list? And Aaron's going to tell me the 2 things, but I do actually want to do one other thing before I forget. So I want to show you those math questions. Like if you have a 1,000-pound horse, you do the 7%. But I want to tell you, like, what if we have 10 pounds of blood? How many pounds did the animal weigh? Okay. Do you guys have a calculator on one of those maybe these guys? Somebody has a calculator on their phone, because when you'll see it, I'm going to write it on a document cam, and I just need somebody to tell me the answer. Let me get this straight up, and I'll show you how to think about this first. And of course, this is always hard. I'm looking this way, and the screen's over that way, so bear with me. Somebody has got that there. Okay, it's pretty straightforward when you have the big part, the whole, going to a small, and in my case, the big square is the whole animal body. The little square is blood weight, okay? So blood weight is a part of the whole. We know it's 7%, so that would be 7 over 100. So the question is, how much blood does a 1,000-pound horse have, or a 100-pound dog? You use the 7 over 100. And why I'm saying 7 over 100, it's better to do that rather than 0.07. And I'll show you why when we set it up. But now, what if I said we have 3 pints of blood, and I'll get some real questions here in a minute. 3 pints of blood, and I'm going to roll that up. And I want you to calculate what the animal weighed when it was alive. Let's say we drain all the blood out of an animal as 3 pints. Your brain automatically should say 3 pounds, right? And so here, you use the inverse. And that's a pretty good diagram to remember. If you're going from the big to small, it's 7/100. If you're going from the small, if you're given, we drained all the blood out of an animal, it's 4 pints. How much did the animal weigh when it was alive? You go 100/7. Okay, so let's do an actual question. And let me do the big to small first. Let's say we have a 1,000 pounds. Now watch how I set this up per horse. Okay, we have a 1,000-pound horse, and I want to end up with how many pounds of blood? The question is pounds of blood. So then you go 7/100, right? And you can calculate that out. We already know that's 70. Now, let's say I want to do pints. How many pints of blood? So I would do that, but then I would say there's 1 pint per 1 pound, and I would calculate that out. You might say, why would I do that? Well, I need to cancel out the pounds, and now if you run that out, it's pints, right? And it's, you might do that mentally because it's a pint to pound to whirl around. Okay, so then, let's say we drain 30 pounds out of an animal, all the blood. Okay, 30 pounds, we drained it all out. That's 100%. Now, and I got one more math question after this too. So Sarah, remind me about the other math question about percent blood loss, okay? Okay, so I got 30 pounds per animal. What did the animal weigh? Somebody calculate that out for me. Who's got a calculator? So you got 30 pounds per animal, but now I want to go from the small, 30 pounds of blood per animal. So 30 pound, maybe we want to do that, blood per animal. And I want to get back to the whole animal. So what is that? Are you calculating that out for me? Here we go.
Unless I calculated wrong, I got 428.
428?
Yeah, 428.57. I don't know how much.
428 pounds. Does that sound about right? Okay. Because 100/7 is what? Is that 12? Real close to a little over 12, right? So if you take 30 times 12, you know you've got 300 plus
14.
Yeah.
Yeah.
That's it, 14?
Yeah, it's 14.29.
Okay.
Yeah.
So 428 looks right. When I do have these questions on the assessment, I'll make sure they kind of come out pretty even, right? So if you know what you're doing, you can pick out the answer, because on the assessment I would never have, let's say if 428 was the right answer, I would never have 425, 428, 430. That's too close, you know what I mean? And it gets around. I would have 428 as the right answer, then I might have 395, 455. See, I'm going to spread away from the right answer. I'm not going to fool you to make, try to have something like that. I know you're using mental math or in the margin. Okay, so then now let's say the question was, how many quarts? That's what I want to do. Okay, so that's how you get up to the big animal. Let me stop right there for that one, okay? Okay, so now, let's say a 1,500-pound horse has lost 12 pints of blood. Okay? So it injured itself. Horses are famous for running into fences and gashing and something. So it's lost 12 pints of blood, you're estimating, or during a surgery. So now the question is, what percent of its blood was lost. Because remember that one article Margaret wrote, was it 30% or less she doesn't worry about IV, right? Fluid replacement, if I remember right. So let's use her number. So this is happening to you, you gotta say, okay, I wonder what percent it is, because, you know, I wanna know if we're in danger or not, okay? So what you have to do then, you know you lost 12, but I wanna know how many total pints in the horse, right? Of blood. So I go, and I'm relying on somebody out there in the audience to calculate. Yeah, oh, sorry, yeah. Keep me in line here because this, I'm looking away from the screen. Okay, and let's see. So, 1,500 times 7 divided by 100, so I guess, really, 15 times 7?
105.
105. So then I would go 12 over 105, and that's my percent. See that?
11%.
11. Okay. I just figured out what percent of blood was lost by that horse. 11%. Am I going to worry? Am I going to go through a transfusion? No. I'm just going to say if it's a healthy horse, we're okay. How's that? And you got to practice because, see, I was doing it, you weren't, right? And, you know, do and understand, all that kind of stuff. So I wanted to do that kind of math question because I can promise 1 or 2 on the assessment. Yeah. Okay, now I think we're ready for to remind me. Hormone half-life. Okay, that can be divided into 2. You can talk about the half-life of a compound. Often it's like this, given like this. I think the Colorado website, and you know, I wish he hyphenated it. He runs it all together, half-life, and sometimes it's hard to read those for me to. Okay, so half-life, however you want to designate it. Now, I'm not going to say a hormone right now, I'm going to say a compound. It's the time it takes for half of the compound to disappear in the animal, be degraded, be excreted. It's, you know, it's half-life, it's half gone. What time does it take for half of the compound to be gone? So let's say I inject 500 milligrams of a hormone into an animal. And I'm going to say the half-life is 2 hours. Can you move it up? Yep, thank you. Yeah, I don't know why this is facing away. See how odd this is? Anyway, and I could have a duplicate on that screen. Okay, so I'm going to say the half-life is 2 hours. Half-life equals 2 hours. And if I inject 500 grams at time 0 milligrams, how many milligrams would be present in the animal, let's say in the blood, after 2 hours? Somebody said 250. Yes, I have to have it. Half it. So there's 250 milligrams left in the animal. And this has big bearing on, like, if you ever inject antibiotics in animals or something, there's a drug withdrawal period. If you're ever familiar with that, if it's an animal that will be used for human food consumption, you know, there's a certain withdrawal period, I guess is the right term. Okay, so then, you know, we're taking blood samples. We injected 500, there's 50 left. Another 2-hour period goes by. What do I want to write up here?
125.
125. That's strange if you're not familiar with it. You want to say 0. Somebody, you know, I usually have volunteers and you want to say 0, but no, it's half again. Just think of this as starting new. Think of the 250 as you just put it in. In 2 hours, it's half gone, 125. So then what's the next 2-hour period? What's that? 60 something, right? 62.5. I'm going to say 63. Okay, so then you keep having it. Now the thing is, you should be able to calculate the other way too. Back the other way. So if I say after 6 hours, we found 63 milligrams in the blood, what was the initial injection? Then you would double it, right, to get back to the time 0. So that's half-life. It's hard to understand. You can talk about testosterone half-life. You can talk about the half-life of any chemical in an animal. And here's the practical thing. Radioactivity has a half-life. Some things have, some radioactivity, when I was working in an endocrine lab, I had a lab, some of it had a half-life of 12 days. But it was like radioactive. But after 120 days, it was just thrown in the regular garbage. Because 10 half-lives equals gone. For all practical purposes. So if you wait 10 half-lives, then it's gone. And that's what Purdue does with the radioactivity. They collect it in all the labs. They collect it, and then they store it someplace, and then it's thrown in the regular garbage after about 10 half-lives. Maybe they go further. But the thing is, so if you inject it into the animal, and we did 500 milligrams, it would be, what, 20 hours we could consider it gone. 10 times the half-life equals gone. That's a very good principle. It's not perfect. Half-life equals gone. By gone, I mean 0, right? Technically not. Technically, you can keep splitting that forever. Okay, anybody have any questions on that? Half-life? I'm doing the document cam. Oh yeah, then it was the Colorado website, right? And then remind me after Colorado, I want to draw the pituitary hypothalamus. Okay. So now, let me get back to our assignment because I want to like brag on this Colorado website. I don't know Richard who does it personally, but I email back and forth when one of the links not working or something. I need some help with his site. He's I can't believe how responsive he is. He'll go out of his way to help me. God bless him. Okay, so here we are, and I don't know if the lights help or not. The first click, hormone 1. Read the short page and click on the 4 core concepts. So by the short page, I mean this page that just comes up. You read that, and then here's the 4 concepts. I didn't assign steroidogenesis, for example. So, but that doesn't mean you can't read it, right? I mean, maybe for your career that might be good. I'm just saying, see, what I do is I do the minimum. I give you the minimum to do. So if you really want a good career, would you always just be doing the minimum? Hopefully not. Anyway, an overview of endocrinology. I mean, this looks kind of cheesy, but he makes just a great point that there's really, to control everything in the body, there's the nervous system, which is coming later, and we're doing the endocrine system now. And then some cell releases a hormone, the blood isn't showing here, but then it binds to a receptor, and then that cell responds. It's a target tissue. There are chemicals in the nervous system too, and sometimes they're both found in the nervous system and the endocrine system. But in this case, here's a neuron, nerve cell body with dendrites around there in one long axon, right? And it's talking to some cell or muscle. And then it's releasing, what's it called when a nerve releases its chemicals into the interstitial space? What are those little chemicals called? Neurotransmitters, okay? So neurotransmitters are released and they diffuse across this cleft into either another nerve or a muscle usually, right? And so he did a good job. And then he's, the videos have more endocrine glands, but there's the location. And of course, most animals don't have ovaries and the testes, but for the drawing, it's good. And then if you ever see pathophysiology, that means physiology gone bad. So we talk about it some here, but you could have a whole course on pathophysiology of the horse, things that go wrong with physiology. Okay? So anyway, very good. And then I guess I could go back and there's other way to get the core, but then there's hormones, receptors, target tissue. And then while I'm thinking about it, oh man, I was very close. Yeah, I'm gonna go just reset my camera. And if you looked for yesterday's lecture, it's not quite up there yet, but it will be. Okay, let's see. Oh, and Taz is sleeping. We got a nap going. That's good. Hopefully she's the only one napping in the class. Okay, so then look at how good this is down here. Oh yeah, and somebody had a question about this yesterday, so that's, excellent. Because he talks about autocrine, paracrine, and endocrine. So very good. An endocrine substance, you might call it a hormone, but you could call it an endocrine substance, is released by some cell, travels in the blood to some other place, target tissue. That other place might be just millimeters away, or it might be 6 feet away, right? It's going to be traveling in the blood. An autocrine substance is released by a cell, and it affects the same cell it came from. Cell, autocrine. So some cells release something and it comes back and affects the same cell. That's an autocrine substance. Paracrine is a great name because para, P-A-R-A, is a prefix meaning near or next to. And a paracrine substance is released by some cell and diffuses to another nearby cell without going into the blood. Okay, a paracrine substance. Yeah, very interesting. So that's how they got their names. Autocrine, paracrine. Sometimes you'll see another crine, I can't remember it right now. It's not very commonly used. Okay. But anyway, excellent. This guy is, bang up. Let me do the other reading. So then the other one is read the short page. That just means whatever comes up. And then do the core concept. So I like how on the last page I didn't show you, but one of the last things he says is about the tyranny of terminology. If you read it, the tyranny of terminology. And he talks about how these hormones are discovered. Somebody names them sometimes, and then near after its discovery, and then years later, it should have been named something else. That's the tyranny of terminology. I like that phrase. I wonder if he made that. So it's confusing because some hormones have two names. I'll give you an example. Growth hormone. What's the other name for growth hormone? Somatotropin. Let's write those down. Because it's a great example of, and so we're stuck with it. So growth hormone or somatotropin. Those are the same thing. So dairy cows are injected with bovine growth hormone to make more milk. Or dairy cows are injected with bovine somatotropin to make more milk. See how somebody could think it's two different hormones? It's not. It's the same. So be careful of that. It's to get into it, it is kind of confusing, and we're trying not to get too confused. Okay. Anyway, his site, it's beautiful. I mean, it's written at your level, and it's free. It's not a $200 textbook. What was the next thing, Sarah? Was it who? Aaron? Oh yeah, drawing of the pituitary gland. Okay, because it's really good to understand that at the base of the brain. Okay, so I'm going to draw. Yeah, I'm on screen. Base of the brain, and if you know from the reading and videos, hypothalamus. And if I scratch a little bit, that's okay because you've got it. So let's say there's the hypothalamus, and then I'm going to make this the anterior pituitary. So I'm going to put that over here because I don't want to mess up the space. So anterior pituitary, then what's this called? posterior pituitary, PP. Okay. Now, what do you think the tissue above the hypothalamus is called? What is, the answer's up there.
The thalamus.
The thalamus. And this is at the base of the brain. It's actually, the pituitary especially, is like in a little depression at the base of the brain. I've taken out cow pituitary glands. If you're a human surgeon and you have to work on the pituitary, you go through the roof of the mouth. Open wide. Surgery. You don't come through the brain or the side. The pituitary is not too far above the roof of the mouth. So you have a retractor for the person and you're sitting there working on the roof of the mouth going up. Okay, so anyway, need another color. Black. You know, or maybe you don't know, thalamus and hypothalamus are all neural tissue. There's neurons. And when we get to neurology, you'll find out there's two types of cells in brain tissue. There's the active neurons, and there's other things that are called glial cells, but we'll get there. But it's all neural. But I was kind of tricky because notice how I left a continuous white space between the posterior pituitary and the hypothalamus? Because some of the cells, the neurons in the hypothalamus, send an axon all the way down to here. So I'm saying the hypothalamus and posterior pituitary are all neural. And some of the cells, some of the neurons send an axon and they end here. That's the axon terminus. That's not the case with the anterior pituitary gland. It is all glands, tissue, cells that release hormones. Plain endocrine cells. Not neural, neural. Now I gotta draw the blood supply. I purposely left red out of it. Now, and then there's better drawings in the videos and in the Colorado site. You know, I'm drawing here. I'm looking to draw a stick figure. So anyway, yeah. And the other thing is, there are some neurons up here that release chemicals up there. So let me try to draw this now. Okay, I've got a blood vessel coming here. And then I'm going to send it this way. Some of these neurons up here release a chemical up here, and sometimes they're called releasing factors. I was talking to somebody the other day about it. In one of the diagrams, it says releasing factors. It could also be labeled releasing hormones. Releasing factors equal releasing hormones. See, this is the tyranny of terminology, isn't it? So we talked about growth hormone. Up here, this is the neuron that releases growth hormone releasing hormone, GHRH. Growth hormone releasing hormone. It's coming from a neuron that terminates near a blood vessel up here in the hypothalamus. So that will make a little speck in the blood vessel. That releasing hormone is made by the hypothalamus, released up here, goes into a capillary system. I'm going to talk about that in a minute. And maybe just, I'm going to do CB for capillary bed. So CB up here, and then there's another CB back here. Because you know capillary beds tend to be leaky. This isn't in the brain per se, so they're still leaky. Remember how I said the blood-brain barrier is tight? They tend to be less leaky, but growth hormone releasing hormone, right? GHRH goes down to this blood vessel and it diffuses out and stimulates these cells. So I'm going to try to pick that. Well, lo and behold, there's still blood flowing, right? So now these cells down here will release growth hormone. Oh, sorry, I pushed the wrong button. They've got all the things right here. Okay, they'll come back. Okay. Okay, so let's go back here. Why is that? No idea where that. Anyway, okay. Thalamus, hypothalamus. Up here, this cell released growth hormone releasing hormone, right? It diffused down to the, I don't know why we're having that action right here, but that's okay. Oh, maybe. That's better. Okay. Then it goes down to here, and these cells release growth hormone, and the growth hormone goes all over. So, let me review that. Because this is the whole thing about releasing factors, releasing hormone. These neurons up here didn't extend down here. They just release their hormone up here. They're a neuron, growth hormone, releasing hormone. Very few molecules come out. So few molecules that you could never measure it in the systemic blood. If you took a blood sample, I've taken blood samples from 2,000-pound bulls by the tail. It's a beautiful place to take a blood sample. I would never find growth hormone releasing hormone in the bull there. It would be so diluted out by that time. So it travels a very short distance, stimulates these cells, and then these cells release growth hormone, and I could find growth hormone in the blood, in the tail of a bull, right? So that's releasing hormones. Then let's make it a little more complicated. Do you know there's another neuron over here? I didn't draw it. That releases GHIH. What's GHIH? Growth hormone inhibiting hormone. Inhibiting factor. So they're like a balance. So some cells are releasing growth hormone inhibiting factor, and they come down here and tell these cells don't make so much growth hormone. So there's a balance on those two. The point is, though, down there, that gets into the systemic circulation. Now let me go to the posterior pituitary, because I haven't drawn the blood vessels there, and I gotta one me out. So here's a blood vessel here, coming by here, and of course, it's the real tissue is better than that. But now this neuron way down here is going to release a chemical into the blood, and that's going to get out to the tail of the cow, the bull. So if it comes from the posterior pituitary, I can find it in the tail of the bull. But if it came from up here, like GHRH up there, too little is released. I would never find it. I would find growth hormone, I would find this, and we'll use this example as oxytocin. So oxytocin can be found in the systemic circulation. So you got to know the difference of that anatomy. The hypothalamus is neural, but sometimes neurons send a long axon to the posterior pituitary. That's not the case for the anterior pituitary. Now, one last thing. Man. Okay. Now this is like a little sidebar, but it's important. See where I said capillary bed? And then I said another capillary bed? That's unusual. You never usually find two capillary beds in a series, because they're basically the same tubing. There's a capillary bed, and then, this is not a capillary bed here, but it is down here. That only happens in 3 places in the body. Because usually when you leave a capillary bed, you go to a venule, right? Remember the last 2 weeks? And then you go to a big vein. Whenever you have 2 capillary beds in a series, where the blood flows through one, and the same blood, without going back to the heart, flows through another capillary bed, that's called a portal system, P-O-R-T-A-L. That's a portal system. And it's amazing. And let me tell you the other 2 tissues that happens. Liver and kidney. But we'll be talking about those separately later. But a portal system is where you Blood goes through a capillary bed, and then it goes through a vessel, and then it goes through another capillary bed without getting back to the heart yet. Just really weird. And someplace in the reading of videos it talks about that, capillary bed. Okay, other questions or comments on that? I think that's a perfect place to end. We'll see you in math. Oh, here's a question. Okay, so I can talk to you. Okay, so see you tomorrow in math 175. Who's bringing in the dog? Okay, what kind of dog is it again? Basset Hound, okay. Yeah, that'll be fun.